This is Unit 2, Part 5. So I talked about this, I believe, earlier. This is a generalized model of a cell, or a diagram of a cell. We're going to go through in more detail each of these components that make up a cell. When you look at the cell, the material between the plasma membrane and the nuclear membrane that surrounds the nucleus is called the cytoplasm. Inside the cytoplasm it's a fluid-based medium with lots of soluble proteins in it, but it also contains membrane-bound organelles. The cytosol is the dissolved part of that cytoplasm, and the cytoplasmic organelles are the membrane-bound structures that are part of the metabolic machinery of the cell. Sometimes there are some inclusions. These are solid material that sometimes is stored inside a cell. And here you just see an overview, a table giving you an overview of the things that we're going to talk about in the next few slides. Okay, so in terms of cytoplasmic organelles, what you have are separate subcellular compartments. These are specialized cellular compartments surrounded by membrane, although there are some that are non-membranous. But essentially you have subcellular organelles that are membrane bound, surrounded, in other words, by a phospholipid bilayer. We will talk a little bit about some non-membrane bound structures that are found in cells. The ones that we're going to talk about, I think some, not all, are listed here. Let's move on. Okay, first let's talk about the mitochondria. These have a double membrane structure. There's an outer membrane and an inner membrane. So the outer membrane is a phospholipid bilayer, and there's another membrane inside the inner membrane, also a phospholipid bilayer. And this is the structure where most of the cell's ATP is produced via aerobic cellular respiration, which probably doesn't mean anything to you, but I'll explain later in the course. Aerobic cellular respiration. Aerobic means it involves oxygen, and it's cellular respiration, a mechanism for generation of energy in the form of ATP. An important point is that mitochondria contain their own DNA and RNA, which is derived from that DNA in terms of the information it contains. So they have their own DNA, which codes for them, and they are in fact self-replicating organelles. So when a cell is dividing into two new daughter cells, the mitochondria will generate more new mitochondria, and it's using DNA inside the mitochondria itself. Here's a diagram on the left and an electron micrograph on the right. An electron micrograph is a photograph taken with an electron microscope, so it's at a very high power. This shows on the left here the outer mitochondrial membrane in yellow, not in yellow, in red here, and the inner membrane in orange. And embedded in the inner membrane are a series of very important enzyme molecules that we're going to talk about. They're important in terms of the process of energy generation. And when you look at the electron micrograph, you can see these shelf-like structures that are formed by the inner mitochondrial membrane, and these are called cristae, or cristae. That's plural. Okay. And again, I remind you that the mitochondria is where most of the ATP is produced in the cell. Now, inside the cell is a non-membrane, non-membranous structure which are called ribosomes. They are granular in structure, granular particles made of two subunits, and they contain protein and ribosomal RNA. This is the site of protein synthesis, other than any protein synthesis that occurs in the mitochondria. This is the site of the vast majority of protein synthesis that occurs in the cells. There are two types of ribosomes. There are ribosomes that are there are ribosomes that are free. They're free-floating ribosomes in the cytoplasm, and they're used for the synthesis of proteins that are soluble, that will remain inside the cell. There are also some membrane-bound ribosomes, and by membrane-bound, I don't mean that they're surrounded so much as they are actually embedded in a membrane structure, and I'll describe that soon enough in the next few slides. But they are attached to or bound into a membrane, and these are involved in the synthesis of proteins that will be either incorporated into membranes, or embedded or integral membranes, or embedded proteins or integral proteins, or what's the term? Proteins associated with the surface membrane, or they are involved in the synthesis of proteins that get exported out of the cell. So two major types, free and membrane bound. Now, the cell contains outside, now we're talking about outside of the nucleus, right? The cell contains a series of membrane sacs, SACS, that are parallel to each other, hanging elongated, interconnected sacs and tubes that enclose a space called the cisternae, but I wouldn't worry about that space called the cisternae. This is continuous with the nuclear membrane. The nuclear membrane is attached to this structure, and the structure, and I'll show that, I believe on the next slide, the structure is called the endoplasmic reticulum. There are two types of endoplasmic reticulum, one that contains ribosomes, and that's called rough endoplasmic reticulum, and some that does not have ribosomes, and that is smooth endoplasmic reticulum. And here you see a diagram showing it. This is the nuclear membrane or nuclear envelope surrounding the nucleus. So in this sphere area is the nucleus. And these in blue, shown in blue, are the membranous endoplasmic reticulum. The ribosomes are colored reds. They're dotting. These are the membrane-bound ribosomes, and therefore it's on that membrane, and therefore that endoplasmic reticulum is rough. And if it doesn't have ribosomes on it, like in this area, then it's smooth endoplasmic reticulum. As I told you, ribosomes are granular structures that have two subunits, a larger subunit and a smaller subunit. So this is a blow-up magnification, and that's what they are showing here with this arrow. That's a typical thing that you see in diagrams describing structures. You see it derives from the original area and is showing as a magnified image of what they're trying to highlight there. So that's a ribosome. Rough endoplasmic reticulum is studded with ribosomes bound to that membrane. It manufactures proteins that will be either embedded in the surface membrane or will be secreted to the outside. So either proteins that are going to be secreted or proteins that will form integral membranes, and also it's also forming the phospholipid for cell membrane, and you'll see how that works. Smooth endoplasmic reticulum, again, a series of interconnected network of membrane-bound sacs or tubules. It's involved typically in catalytic reactions, enzyme catalyzed reactions, and it's found in cells in a variety of areas of the body. And here's just a series of examples. I wouldn't worry about the details here, but you should know that it's important in catalytic reactions in cells in different parts of the body. Now, outside, further outward towards the plasma membrane, away from the rough endoplasmic reticulum, is the Golgi apparatus, or the Golgi network. Sometimes it's called a Golgi network, the Golgi apparatus. So here you see in blue the endoplasmic reticulum. It's rough endoplasmic reticulum because it has ribosomes associated with it, and you have the ribosomes are synthesizing protein based on information coming from the nucleus of the cell. The nucleus of the cell, of course, contains DNA, and the DNA has genes in it. So there are genes, and each gene in the DNA codes for a different protein. And that information coded for in a particular gene, if the gene gets transcribed, the information is transferred into a newly formed messenger RNA molecule, which leaves the nucleus and enters into the rough endoplasmic reticulum and goes to ribosomes. It's at the ribosome that the messenger RNA, I'm going to write that out for you. So information from DNA, a gene in the DNA, that information is transcribed into messenger RNA. A messenger, if a gene is turned on, activated, then the information in that gene is used to form messenger RNA. And that RNA then leaves the nucleus and goes to the endoplasmic reticulum, to ribosomes in the endoplasmic reticulum, where it is translated. The information in the messenger RNA is used to form protein, a specific protein. So that process is called translation, and it's used to form a specific protein. So information goes from the DNA to messenger RNA to protein as it moves from the nucleus into the rough endoplasmic reticulum to the ribosome, where it forms into a particular protein. And the proteins are packaged. The newly formed proteins, as you can see here, are being packaged. They're represented in the diagram as these blue spheres. I don't know how well you can see them, or here. And they're being packaged into membrane-bound vesicles that are budding off of the endoplasmic reticulum. So here you have in this space, the space inside the rough endoplasmic reticulum. Remember, I told you the space is called a cisterna. And in this space here and in here, inside the rough endoplasmic reticulum, proteins are being secreted into that space as they're being made at the ribosomes, and you get them packaged into these membrane-bound vesicles. And there's a vesicle. And the vesicles move from the endoplasmic reticulum and fuse with the Golgi apparatus. And here you see that process happening right there. You get fusion of the vesicle with the Golgi apparatus, and therefore with that fusion you have the protein that's inside the vesicle entering into the space inside the Golgi apparatus. Inside the Golgi apparatus, proteins are processed further. Many proteins that are made undergo post-translational modifications. They're cut shorter or cleaved quite often in different places, so you can have multiple. From an original protein produced, you could have it processed into several different other types of proteins because it's chopped up into different portions. It not only gets cut, but sometimes different parts will join with each other. There may be an addition of a carbohydrate portion to the protein so that it becomes a glycoprotein. All that can happen inside the Golgi apparatus. So protein processing is occurring in this space, and the proteins are undergoing the final modifications and are packaged into vesicles as shown here. So you're getting that vesicle filled with the newly synthesized proteins, and it gets released as a secretory vesicle, migrates to the cell surface membrane. There's the membrane, the plasma membrane or the cell membrane, and it's fusing with the cell membrane. So this is a process of exocytosis, and it's secretion of a newly synthesized protein by the cell using the process of exocytosis, of a type of vesicular transport, which you've learned about, requiring energy, and then therefore is an active type of transport mechanism. Some proteins are packaged into vesicles, but the vesicles stay inside the cell. An example of that would be proteins which stay inside another type of organelle called a lysosome. Lysosomes contain hydrolytic enzymes. Remember, hydrolytic enzymes are used to break down the 4 major types of molecules that are found in living things. Proteins, carbohydrates or polysaccharides, lipids, and nucleic acids. So if the protein is hydrolytic enzymes, sometimes they're packaged into vesicle, which is called a lysosome. Now, what happens, you already learned about phagosome. I told you about a phagosome. That's a vesicle that's produced during phagocytosis. So if some solid object is brought into the cell using endocytosis, and if the type of endocytosis is phagocytosis, then you get what's called a formation of a phagosome, a membrane-bound phagocytic vesicle, right? Now, phagosomes can fuse with lysosomes. So a phagosome can fuse with a lysosome, and that's what's being shown here in the diagram of a phagosome here fusing with a lysosome. And when the two membrane-bound organelles fuse together, you get the release of the hydrolytic enzymes into the phagosome. At that point, the phagosome is called a phago or phago, but I use phagolysosome. Okay, so with phagocytosis, you get formation of a phagosome, and the phagosome will then fuse with a lysosome to form a. Oh, I spelled it wrong. Sorry. I'm going to take this off here. Didn't have enough space for it. To form a phagolysosome. Following fusion, you have formation of a phagolysosome. And what happens is inside the phagolysosome, you get digestion, hydrolytic enzymes acting on whatever solid thing the phagocytic cell took in its phagocytic vesicle, its phagosome. Phagolysosome. P-H-A-G-O-L-Y-S-O-S-O-M-E, a phagolysosome. That's a very important process because you cannot have release of hydrolytic enzymes into the cell, free in the cell itself. Otherwise, the proteins, carbohydrates, lipids, and nucleic acids inside the cell would get broken down. It has to be compartmentalized inside a lysosome, and the enzymes then get released into the compartment of a phagosome, the structure then being called a phagolysosome. And so the digestion process, the hydrolytic digestion process mediated by the catalytic effect of the hydrolytic enzymes, is all occurring inside the phagolysosome. The other type of process shown here I'm not going to talk about, except to point, well, I will point out that you can get, you should understand that whatever phospholipid bilayer membrane is being formed in the endoplasmic reticulum, right? Whatever membrane is getting formed with the formation of the endoplasmic reticulum membrane will also be exported with the export of the vesicle. I mean, it's bound by membrane. The membrane is fusing with the Golgi apparatus, so it's adding phospholipid to the Golgi apparatus. Then the Golgi apparatus was releasing some membrane, phospholipid membrane, with vesicles it's forming, and those migrate to the surface, and so new plasma membrane, in fact, is coming essentially from the endoplasmic reticulum, going to the Golgi apparatus, and then going up to the plasma membrane or cell membrane. Okay, the Golgi apparatus, as I said, similar in shape to the endoplasmic reticulum, stacked, flattened membranous sacs involved in modification and packaging of proteins, concentration of proteins, addition of carbohydrate groups to proteins, cleaving, chopping them up. The transport of vesicles exported from the endoplasmic reticulum fuse with what's called the cis face of the Golgi apparatus. Cis is the same, so it's on, facing the side facing the endoplasmic reticulum is where they fuse on the what's called the cis face of the Golgi apparatus. Then the proteins pass through and get modified and are exported as vesicles, transport vesicles from the trans face. I said transport vesicles, but they're really secretory vesicles. So it's exported now from the trans face, the opposite face. Trans means across, opposite face, and the secretory vesicles leave that trans face and go up to the, well, they go wherever they're designated to be or needed. If they contain hydrolytic enzymes, they're going to go towards forming lysosomes, or they may be carrying secretory molecules that move up to the plasma membrane, fuse with the cell membrane, and release proteins there. So here you see a diagram just showing the Golgi apparatus. The endoplasmic reticulum would be back here in this area. Here's a transport vesicle coming from the rough endoplasmic reticulum and fusing with the Golgi apparatus. It is fusing with the cis face. Remember, it's fusing here with the cis face of the Golgi apparatus and binding to and emptying its contents into the protein contents into the Golgi apparatus, where it undergoes processing and modifications, and then it gets exported at the trans face inside secretory vesicles that move up to the surface and get secreted or become lysosomes or some other structure. Lysosomes, as I said, contain digestive enzymes, hydrolytic enzymes. They're involved in digestion of extracellular molecules that get phagocytosed into a cell. Could be bacteria or viruses, any solid material or some molecules sometimes that come in. They become degraded by the digestive enzymes. They come inside an endosome or a phagosome, and then you get fusion with the lysosomes. You get the enzymes coming in and breaking them down. These are just some examples of lysosome actions in different types of cells that they can act. Secretory lysosomes are found in some cells. They're quite important in certain cells, but let's move on. I'm not going to dwell on that part. Here you see an electron micrograph of some lysosomes inside a cell. This, remember, is our generalized diagram of the cell. They've taken all the color and detail out and are just highlighting the lysosomes. But you should easily be able to look at it and realize, okay, can I identify the different parts? This is the nucleus here and the nuclear membrane surrounding it. In the middle is the nucleolus. There's the nuclear membrane. Immediately outside it is the endoplasmic reticulum, and further outside from that is the Golgi apparatus, shown down here mostly. And here are some mitochondria, and here are some lysosomes. Okay, so to continue with what we were talking about, the whole system of production of proteins production, storage, export, all of that, or breakdown of things that are taken in, including bacteria, for example, or viruses that are taken into a phagolysosome. All that system is called the endomembrane system, and it includes the nuclear envelope, because if a gene in the DNA is turned on and is transcribed to a messenger RNA, then it will move out of the nucleus through the nuclear envelope into the endoplasmic reticulum, and will interact with ribosomes there, and the information is translated into proteins that are produced. And that whole system that I just described with rough endoplasmic reticulum and transport vesicles and cis face and the trans face of the Golgi apparatus, receiving and sending out proteins that were processed, all of that is the endomembrane system. And here you see what I'm talking about. So there are genes inside the DNA that's found inside the nucleus, and they can exit. If they're transcribed to messenger RNA, the messenger RNA can exit. You see the space of, this is supposed to be a pore here, and it's continuous with the internal space, the cristae of the rough endoplasmic reticulum. And proteins are produced and either exported into lysosomes or go into vesicles. There's no vesicle shown here, but a vesicle that will fuse, say, with the cis face of the Golgi apparatus. Then the protein is modified, undergoes post-translational modification inside the Golgi apparatus, and is packaged into transport vesicles that can be secreted to the outside. Right? It can fuse with the plasma membrane and allow the protein to be released to the outside. Okay, another type of membrane-bound organelle found inside the cytoplasm of cells are peroxisomes. And these peroxisomes contain enzymes, classes of enzymes that are called oxidases and catalases, and these are these enzymes are able to break down certain types of toxins that are sometimes found in cells. The toxins are called free radicals. They're often called oxygen-free radicals. They are produced by cells, the normal metabolism of cells, and are highly reactive and can injure cells, so they have to be neutralized. The cells also contain a skeleton. It's made up of fibrous proteins. We talked about fibrous proteins earlier in a previous unit. These are insoluble fibrous proteins made up of a lot of repetitive secondary structure, typically very long molecules. And the skeleton of the cell does have a framework. And it's made up of three major kinds of cytoskeleton molecules. There are structures. There are microtubules, microfilaments, and intermediate filaments. This diagram on the left is a diagram, and on the right are photographs taken with a microscope. On the left, you see microfilament is made up of repeating units of a protein called actin. And you can see that it's made up of alpha helices of long alpha helices of proteins. It's cut short here and here, but it could be much longer. Made up of individual actin subunits. Intermediate filaments, again, made up of these helical structures, and microtubules are made up of the protein tubulin, so they're tubulin subunits, and again in an alpha helical pattern. These photomicrographs, I should point out to you, are made using antibody that is specific for microfilaments or intermediate filaments or microtubules, and the antibody has a fluorescent dye bound to it. So antibody is put on the cells, incubated with the cells for a while, then the cells everything's washed. If the antibody did not bind to anything on the cells or in the cells, then it gets washed away, and there won't be any fluorescence when it's put in a fluorescent microscope. But in this case, you can see that the antibody that has a green fluorescent dye has bound in here, all throughout the cytoplasm, all through this area that's now green. That's one cell. All of that's the cytoplasm of a single cell. The orange part is a non-specific fluorescent dye that can bind to DNA, and so the nucleus shows up as orange. But the rest of it is antibody against actin with a green fluorescent dye bound to it. And in this case, the next one down, the second one, B, is cell with, this is one or two cells. It's hard to tell. I would say at least two cells there with antibody to intermediate filaments. And then again, using a non-specific dye for the nuclei. You see in the third case, C staining for microtubulin or microtubule structure. The nuclei of each of the cells are stained fluorescent blue. That's a non-specific stain for DNA. And the microtubules are stained fluorescent green using an antibody bound with a molecule that's fluorescent under UV light. So microtubules are long hollow tubes made of the protein tubulin, repeating units of tubulin. They determine the overall shape of the cell and also the distribution of organelles within the cell. That's microtubules. So the protein is tubulin, and this is the function. And microfilaments are made of the protein actin. Microfilaments were shown in A on the previous photomicrograph, and microtubules were shown in C, I believe. So you have these strands of actin protein that make up many repeating units, subunits of actin, making up microfilaments. It's attached to the cytoplasmic side of the plasma membrane and stretches across, bracing and strengthening the cell surface. They also attach to cellular adhesion molecules that are embedded in the membrane, so they can help in the function of endocytosis and exocytosis. And the third type, which was B in the photomicrographs, were intermediate filaments, again insoluble, long protein fibers. They resist forces that pull on the cell, and they form the desmosomes. If you remember the junctional structures called desmosomes. Okay, also found within cells and not membrane bound are these structures called centrioles. Centrioles are barrel shaped. They're located typically near the nucleus. You have these 9 triplet arrangement of microtubules in them. They are important in phagocytosis, and they are also found at the base of where cilia or flagella enter into the cell. Right at the base of them, you'll find a centriole. And here you see magnified this centriole arrangement of microtubules. It's 1, 2, 3, 4, 5, 6, 7, 8, 9 triplet structure. The cilia are, as I believe we've mentioned at some point, no, maybe not. Cilia are extensions away from the surface of the cell that cells use to move things along. You find them in the upper respiratory tract, in the trachea and in the bronchi, larger bronchi, and they're continually moving to push mucus upwards. So any dust that might be caught in the mucus on the surface of your airways will then get pushed up and out of the airway. And they look like this in cross section. Again, this a series of 9 pairs in this case. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 2 in the center. So 9 pairs and a pair in the center. Again, microtubules. So this is a classic arrangement, and there's the centriole with 9 triplet pairs of microtubules. Here you see the action of the cilia. They starting at 1, it beats forward to the right in 2, 3, and 4, and then slowly comes back in 5, 6, and 7. So always beating in one direction. And here you see what I was talking about, where you have a ciliated epithelial cell lining the airway, and you have mucus, of course, because there are many cells that secrete mucus in your airway, upper airways, and the cilia are always beating and moving this upwards out of the airway so that it doesn't get clogged up with mucus filled with dust particles. The nucleus. The nucleus is present in every cell except for the only cell that doesn't contain a nucleus are the mature red blood cells. Otherwise, every cell contains a nucleus, and it is surrounded by a nuclear envelope or nuclear membrane. And inside is a nucleolus The plural for nucleolus is nucleoli, but inside is a nucleolus. The chromatin is the DNA material. And you have also quite a bit of protein associated with that. Inside the nucleus is the DNA contains all the information for that cell, for that organism. So all the genes are present in the nucleus, contains the whole genetic library for all the cellular proteins and structures and functions. And here you see a diagram showing that nucleolus. It has one unique thing that a lot of people neglect to point out when they're teaching it, I think, and that is that it has a double membrane. Okay, it's not a single membrane. The nuclear envelope or nuclear membrane is a double phospholipid bilayer. There are nuclear pores, and as I told you before, those pores extend into the endoplasmic reticulum. Here you see the space inside the endoplasmic reticulum called the cisternae. And remember that inside the nucleus you have genes that code for different proteins, and the information can get transcribed from the gene and to form messenger RNA, which will contain the information and exit out into the can go out into the rough endoplasmic reticulum, where that messenger RNA then goes to the ribosomes and is used, the information is used to synthesize protein. It is a selectively permeable membrane, a double membrane, a double phospholipid bilayer, has pores. The material inside is a jelly-like substance called nucleoplasm. The outer membrane is continuous with the rough endoplasmic reticulum and may have its own ribosomes on it. The inner membrane, I wouldn't worry about this. And large molecules can get transported out from the nucleus, in or out of the nucleus. The nucleoli is the site of production of substance called ribosomal RNA. And ribosomal RNA forms part of the ribosome. The nucleus itself, of course, the main thing is that it contains DNA, and the DNA is arranged as chromatin. What chromatin is are thread-like strands, long strands. Chromosomes are made of chromatin, and those are long, thin strands of DNA coiled around histone, a protein called histone. There are subunits to the chromatin called nucleosomes. They're always the same in size, all along, formed by the DNA and the protein histone. That's the end of Unit 2, Part 5, and the last slide in Unit 2.